Time for discussion. We will go ahead and get started. So it is my pleasure to introduce Dr. Riaz Gillani and Dr. Ryan Collins, both from Harvard. Dr. Riaz Gillani grew up in Fredericksburg, Virginia, and subsequently studied biomedical engineering at Brown University. He completed medical school at the Johns Hopkins University School of Medicine, pediatrics residency in the Boston Combined Residency Program, and Pediatric Hematology Oncology Fellowship at the Dana-Farber Cancer Institute and Boston Children's Hospital. As a postdoctoral researcher, he trained under the mentorship of Dr. Van Allen and Dr. Katie Janeway, publishing research on somatic structural variants in oncogenesis and germline risk factors for pediatric solid tumors. Clinically, Dr. Gillani cares for pediatric patients in solid tumors and hematologic malignancies. He is the principal investigator of a research laboratory focused on computational pediatric cancer research based at the Dana-Farber Cancer Institute. And his co-presenter, Dr. Ryan Collins, is an instructor at the Dana-Farber Cancer Institute, where his research focuses on discovering new genetic risk factors for cancer using large-scale genomic data science approaches. Dr. Collins completed his PhD at Harvard under the mentorship of Dr. Michael Tolkowski. His dissertation research involved the development and application of new methods for the analysis of structural variation in tens of thousands of human genomes as part of the Genome Aggregation Database, or GNOMAD project, as well as across various pediatric and adult onset Mendelian diseases. Ryan grew up in New Hampshire, holds a bachelor's degree in genetics from Dartmouth College, and lives in the Boston area with his wife, 2 kids, and their dog. And Doctors Gillani and Collins will present work from their recent publication titled Rare Germline Structural Variants, Increased Risk for Pediatric Solid Tumors. They will give us about a 30-minute presentation, and then we should have time for questions and discussion at the end. So Doctors Gillani and Collins, I will hand it over to you, and thank you so much for being here.
Thanks so much for that great introduction, Dr. Market, and it's a pleasure to be here. Just want to make sure you can see the screen in presenter mode. Okay, perfect. So we'll just go ahead and get started. So we will be talking about structural variants today, but kind of more broadly, we'll be talking about rare germline variants and how they help us understand central oncogenic programs in pediatric solid tumors. And so with that, I'll just go ahead and get started. These are our disclosures. And we'll start with a little bit of background about rare pathogenic germline variants in pediatric solid tumors more generally. And then I'll hand it over to Ryan to talk about germline structural variants in pediatric solid tumors. And then we'll conclude. So I think just to set the stage, it's important to mention that we think about the germline and tumor as part of a spectrum. We're thinking about both as important to fully understand oncogenesis. One truly informs the other in terms of variants in the germline, really kind of leading to different patterns of mutations and rearrangements in the tumors. And today we'll be focused mostly on the germline side of the spectrum. And a central question that drives a lot of our work is, why does pediatric cancer happen? And I think that is probably true for many folks on this call. And then kind of a corollary question to that is, how can we use this knowledge to do better for our patients? And so as this audience is well aware, the mortality and morbidity associated with pediatric cancer remains unacceptably high. And this is especially true for the third of pediatric cancers that are extracranial solid tumors. And so the thinking is, if we can understand the earliest biology that leads to the development of these diseases, we can hopefully be more targeted and thoughtful in terms of how we treat our patients. And the literature over the last decade has really kind of pointed to this statistic of about 10% of pediatric cancers being due to germline risk factors. However, I think a lot of us intuit that as data sets grow, as we broaden where and how we look, we're inevitably going to learn more and find more. And as we're thinking about germline risk factors, I think a framework that's really important to ground us is this common variant, rare variant framework, where common variants by definition occur more in the population, but have lower effect sizes. Rare variants, less frequent, again, by definition, but have higher effect sizes and are often more interpretable based on being tied to specific genes. And so some of the work that we're going to talk about in structural variants is actually informed by some threads that came out of previous work focused on rare pathogenic germline variants and pediatric sarcoma subtypes. And just to share a little bit of background from that work, what we did in this study from 2022 is we analyzed data from different consortia, including St. Jude Cloud, Gabrielle Millikids First, as well as the Framingham cohort for cancer-free controls, and really just tried to parse out different patterns of rare pathogenic germline variants and different pediatric sarcoma subtypes. Specifically, most representative of osteosarcoma, rhabdomyosarcoma, and Ewing sarcoma. And what we found was that germline TP53 variants were unequivocally enriched across the pan sarcoma cohort, perhaps not unsurprisingly. We also saw signals in other implicated genes or genes that have been previously implicated, like PTPN11 and DICER1. However, what emerged when we looked at specific histologies were really different patterns in these different subtypes of pediatric sarcomas. And so in osteosarcoma, we recapitulated the signal in TP53, as well as other described signals in genes like RECQL4 and RB1. In rhabdomyosarcoma, again, saw signals in TP53, as well as other previously reported genes, BRCA2 and DICER1. And the landscape in Ewing sarcoma was overall relatively flat, with the exception of an enrichment signal in this Fanconi anemia gene, FANC-C. And so without getting into all the details of that study, we did replicate that signal in another independent cohort, and really kind of interpreted this as recurrent enrichment of heterozygous pathogenic germline variants in FANC-C and other DNA damage genes as well, that we were finding in the study, provided evidence for the role of these genes in increasing risk for some individuals with Ewing sarcoma. And this is possibly through the mechanism of an increased risk for translocation, which universally occurs in this disease and the tumor itself. And so with that background on kind of differential patterns of rare pathogenic germline variants, I'm going to hand it over to Ryan to talk more about our study in germline structural variation in pediatric solid tumors.
Thanks, Riaz. So yeah, picking up on this thread of previous work having really focused largely on short variants in the germline. So these are SNVs and indels that typically involve no more than, say, 50 nucleotides of DNA. There's sort of another entire class or family of genomic rearrangements that folks in the cancer community, specifically in the somatic genome space, are very, very familiar with. And namely, this is structural variants. So just definitionally, we're talking about any rearrangement of genomic DNA that involves more than 50 nucleotides. In the somatic space, I don't need to make the point that these types of rearrangements are incredibly important, both for the onset and progression and response to treatment in a variety of different cancers, including pediatric cancers. But these structural variants actually also occur as standing variation and rare variants, common variants, just like Riaz was saying earlier, in the general population in the germline. So every germline genome, including everyone on this call, has approximately 20 to 30,000 of these germline structural variants. And one thing that's important to note about germline structural variants is that they are incredibly mutationally diverse. And so we don't see in the germline genome quite as much complex rearrangement and whole chromosome arm swaps and aneuploidies as one might see in somatic tumor genome. However, we do see tens of thousands of smaller deletions, duplications, mobile element insertions, inversions, and several hundred complex structural variants per genome, although universally these are typically smaller in size than you would see in a somatic tumor genome. But because of technical challenges associated with these germline structural variants, they have been historically understudied in most disease contexts, including in part pediatric solid tumors, which is going to be the main focus of our sort of new research that we'll be presenting today. And I just want to emphasize from a technical perspective, one might wonder why this is. And I think it boils down to really 2 points. One is that to solve the problem of germline structural variant analysis comprehensively in human genomes, you really need genome sequencing. And so here we're going to be talking about short read genome sequencing, which is sort of a conventional Illumina technology that I'm sure many of you are very familiar with. But even better would be in the future, long read sequencing, such as PacBio or Oxford Nanopore. These technologies do a better job of resolving especially small and repetitive structural variants than short reads. But even, short reads alone can really make massive headways into this problem. And heretofore, it's been pretty expensive, especially to generate such data sets on rare diseases like these pediatric malignancies. And then the second component to emphasize here is that, and I won't get into the technical details, but technically this problem has stymied the field of human genetics, population genetics, and in particular for decades. Since the onset of next generation sequencing with the exomes in 2011, this has been a really, really hard nut to crack. And I think just to boil the problem down into a few sort of snippets so you can understand, it's that the dimensionality of this problem is really high. Namely, we want to do this analysis in human populations. So you're talking about thousands of genomes. This is terabytes or petabytes of data. It would not fit on one computer. You really have to use cloud computing to solve this problem, which makes it more complicated. And then in parallel, for each of those 1,000 human genomes, you need to be considering multiple different forms of evidence present in the raw sequencing reads themselves, as well as looking for all of those different classes and subtypes of structural variants that might occur, deletions, duplications, insertions, inversions, and whatnot. And so I hope you can appreciate that just the dimensionality of this problem makes computational analysis quite challenging. In my past life over at Mass General Hospital and the Broad Institute, when I was completing my PhD with Dr. Michael Talkowski and members of the Broad Data Sciences platform, we spent about 5 years developing this method, which is now available for public use on the cloud, called GATK-SV. And shown here is a schematic of sort of how the pipeline works. And I won't go into the technical details to focus on the biological insights from this tool applied to pediatric cancers, but this tool we designed specifically to solve this problem of comprehensive germline structural variant detection in large cohorts. And by large, I mean, at least 1,000 samples of germline short read genome sequencing. So the type of thing you would get from an Illumina sequencer. I do want to highlight just a couple features so you understand how this pipeline is working. And so the first thing is that no one method that exists out there in the structural variant ecosystem right now can capture all types of structural variants since they're so mutationally diverse. So we maximize our sensitivity by running many different algorithms, accepting that this will have a very high false positive rate. We then go back and integrate these calls across the different individual algorithms and statistically re-adjudicate the raw sequencing evidence, like I was mentioning in the previous slide, for every sample at every predicted breakpoint present in our cohort. We then use machine learning and essentially, formally a suite of random forest filters to tease apart which are likely false positive germline structural variants and which are likely true positives. And then the back half of this pipeline is really sort of Mendelian first principles, population genetic refinements of this call set. So we assign a biallelic genotype to every sample and every structural variant, at least biallelic for autosomes at least. We annotate predicted coding and non-coding regulatory element disruptions. And other such, we resolve sex chromosome differences between, XX and XY individuals and so on and so forth. So at the end of the day, what comes out of this method, GATK-SV, is a single call set, which has all structural variants that were detected in your cohort with a genotype in every single individual in your cohort. So with this tool in hand, Riaz and I got together, several years back and started talking about, was there an opportunity to apply this method, this population genetic method, to pediatric cancer cases and cancer-free adult controls to ask questions about, this central hypothesis driving our work, which is that germline structural variants may help to explain some fraction of heretofore unexplained sporadic pediatric cancer cases. And so all of this culminated in this study that just came out, 4 weeks ago, something like that in Science. And the graphical abstract is shown here just to give you an overview of sort of what we did and some of the key findings. We'll be walking through all of this, so don't worry about digesting all of this right now. But essentially, we undertook a population-based analysis across 3 pediatric extracranial solid tumor histologies, Ewing sarcoma, neuroblastoma, and osteosarcoma, compared those genomes to about 6,500 adult population controls. These were, to the best of our knowledge, cancer-free at time of enrollment in the studies that we, analyzed the data from, as well as about 1,000 of the parents of these children with these pediatric malignancies, and the parents were, again, to the best of our knowledge, cancer-free. We applied the tool that I mentioned on the previous slide, and then performed a variety of statistical tests that we will talk through in the subsequent slides to help understand if there are any pockets of germline structural variants that are different in rate between the affected children and unaffected adult controls. More specifically, this is just essentially expanding on what I described in the last slide, which is that our overall cohort, after being very strict with quality control to ensure that we only have the highest quality germline sequencing in this data set, included about 1,700 or 1,800 affected children, about 1,000 of their parents, and then about 6,700 unrelated cancer-free adults. And these samples were drawn from a variety of different projects, which you can see here. By applying this pipeline, GATK-SV, to the cohort of about 9,500 germline genomes that we just talked about on the last slide, we captured 284 distinct structural variants present in 1 or more samples in the call set. And one important thing to note here is that 84% of these are rare variants, meaning that they appeared in less than 1% of all individuals in our study. So this is quite a bolus of rare genetic variation that hasn't yet been explored at sequence resolution in these diseases. But looking away from rare variants, most of the contribution for genetic variation in any one individual germline genome is going to be from common variation. And so when we look at the total count of germline structural variants per genome-wide, so this is all structural variants in cancer-free controls shown in gray, or the 3 pediatric cancer histologies shown in the 3 different colors, we saw no really striking differences in the total number of structural variants. So if there was a signal to be found, we reasoned that it would be concentrated in specific pockets of structural variants, perhaps disrupting genes or other, features. So one of the first places that we looked, outside of looking genome-wide, we then started to zoom in on what we felt were some of the most likely subsets of structural variants that might harbor predisposition or risk for pediatric cancers. And one of the first places we looked was in very large germline structural variants, and specifically copy number variants, defined here as any germline deletion or duplication involving more than 1 million base pairs. This is, of course, very small in a somatic genome space, I realize, but in the germline genome, these are incredibly rare. It's less than 1 or 2% of the general population will carry such an event. And the reason we started here is because before working on pediatric cancers with Riaz, I spent about a decade working on neurodevelopmental disorders and pediatric developmental disorders. And we can say from, decades of work in that space that these large, rare germline copy number variants are the single most frequent genetic cause of abnormal human neurodevelopment. And so because of that, we reasoned, well, these are a very important predisposition factor for altered pediatric neurodevelopment. We're not looking at sort of a constitutional or an inherited disease in this context of pediatric cancers, but it's still a relatively severe pediatric onset phenotype. And so perhaps there's risk that's coming from these very large rare structural variants in pediatric cancers. And so shown on the left here, the leftmost panel, is the sort of primary result, which is we essentially tallied the rate of rare large copy number variants in all 3 pediatric cancer histologies compared to ancestry-matched and sex-matched control individuals shown in gray. And you can see for all 3 histologies, we observed about a 2-to-1 ratio of the rate of these large, rare germline structural variants. Notably, and I won't focus on this, we did not see this for balanced structural variants, meaning large inversions, reciprocal translocations, things of that nature that don't result in gross DNA dosage changes. So interestingly, this signal was concentrated exclusively in large deletions and duplications. We then did a sensitivity analysis shown in the center to show that this signal really was most prominent, where the case and control difference became the greatest for very, very large events. But you do see, increasingly modest difference between cases and controls below our arbitrary threshold of 1 million nucleotides. This holds at 500 kilobases. This holds at 50 kilobases, just with smaller and smaller effect sizes. And then where things got a bit surprising was we then wanted to explore patterns related to sex difference in risk factors because, as I'm sure many members of this call are aware, there is a, slight but pretty robust epidemiologic observation of a slight bias towards males being diagnosed with pediatric cancers, and specifically all three of these histologies in this study slightly more frequently than females. And so male and female here, we're talking about germline karyotype of XY or XX specifically. And so we cross-analyzed this large rare structural variant result by only comparing XY cases and controls and only comparing XX cases and controls, and found, to our surprise, that the entirety of the enrichment, statistically speaking, the entirety of the risk contributed by these large rare copy number variants in the germline was concentrated exclusively in XY cases versus controls, with no significant difference found in XX cases versus controls. So I think obviously this is a bit of a surprising result. It is encouraging that it matches, reported epidemiologic, observations, but the mechanism here, I think, is something that we are still certainly very curious to understand, and is going to be a focus of, further follow-up, and we would love to talk to anyone who's, interested in teasing this apart.
And so zooming out from large structural variants, we also sought to kind of understand what patterns of enrichment we would see in rare coding variants more generally. And what we did observe was that there was enrichment in pediatric cancer patients in rare gene disruptive germline SVs, both loss of function structural variants as well as copy gain variants. And we wanted to evaluate these signals more systematically for these coding structural variants. And so we employed a framework called a category-wide association study, or CWAS framework, to systematically evaluate these signals in rare structural variants impacting coding regions. And just to give you a sense of kind of how what goes into the CWAS, we basically use various filters characterizing the structural variants themselves. So what kind of structural variant is this? What's the predicted impact? As well as the genes that they impact. So are structural variants impacting DNA damage repair genes? Are they impacting mutationally constrained genes, or genes that are expressed in the tissue of origin? To enumerate somewhat exhaustively all the categories that we can think of variants. And we see about 700 categories for Ewing sarcoma and neuroblastoma that have a minimum threshold in terms of structural variants in them to evaluate for enrichment in cases or controls. And so to make this a little bit more explicit, an example of a category here would be singleton loss of function deletions in unconstrained DNA damage repair genes expressed in the tissue of origin for neuroblastoma. And so with that said, this is what we found as far as the results of the CWAS study when evaluating coding variants. And so the results for neuroblastoma are in the volcano plot on the left. The results for Ewing sarcoma are in the volcano plot on the right. The x-axis is the effect size, and the y-axis is the p-value. And each dot is one of those categories. And so what we see kind of more generally is that in neuroblastoma, the signals are stronger. Neuroblastoma relative to Ewing sarcoma has an earlier age of onset. And then when we start to look at what these categories that harbor signals and enrichment, whether they be at Bonferroni significance or nominal significance are, we start to see some interesting patterns emerge. So in neuroblastoma, in addition to just singletons, so the rarest of rare, loss of function structural variants, we see structural variants specifically impacting mutationally constrained genes. We also see structural variants impacting double-strand break DNA repair genes that are enriched in cases relative to controls. And this parallels some short variant signals that have been reported in neuroblastoma before, specifically involving FOX2B as far as constrained genes go, BARD1 as far as double-strand break DNA repair genes go. So it's interesting to see some of these same themes coming out in the structural variant side of things. In Ewing sarcoma, as I said, the signals are more modest, but in terms of some interpretable categories that emerge, we see structural variants impacting DNA damage repair genes as well, again, paralleling some of the signals we were seeing in the short variant space. Pivoting to more clinically interpretable genes, we sought to ask what rare germline structural variants disrupt known cancer genes. And so, as you can see here, there were several examples that emerged as far as genes that many of us are familiar with. So in the context of Ewing sarcoma, some specific DNA damage repair genes involved PALP2, the Fanconi anemia gene, FANK-A, both of which harbor deletions. And then in neuroblastoma, as I mentioned, there have been recurrent reports of signals and established cancer predisposition risk associated with FOX2B and BARD1, and we saw structural variants impacting these genes as well.
Then we wanted to further characterize the functional consequences of these coding structural variants, and this was really work led by Jet Crowdis. He's a phenomenally talented MD candidate here at Harvard that Riaz and I had a chance to work with on this study. And so what Jet did was to try and contextualize the effects of these structural variants that disrupt genes on the gene expression levels, both in adult tissues, healthy tissues, as well as in the tumors, matched tumors from the children whose genomes were analyzed in this study. And so shown on the left is one really sort of sensible finding that was really encouraging to see is that in general, the genes that are being disrupted by these rare structural variants in the germline genomes of children with pediatric cancers tend to be, on average, more highly expressed in, healthy tissues than the genes that are disrupted in healthy cancer-free adult controls. But not only are they more highly expressed in general, but they tend to be most highly expressed and particularly enriched in the tissue of origin that was, for the data available to us, was the best match for the putative, cell type of origin or tissue of origin for the disease. And this is, namely, we're using proxies of adrenal gland tissue for neuroblastoma and skeletal muscle for Ewing sarcoma. And certainly this is something that we would love to dig into with perhaps single cell expression data in the future. But even at the sort of tissue level expression data that we had available from healthy adult donors from the GTEx project, we see really nice correlations between high expression, specifically in the tissue of origin or proxy for tissue of origin, and those two diseases. Not only did these structural variants have detectable effects on gene expression in normal adult tissues, but actually in the matched tumors, Jet was able to show on the right here that even though our sample size was limited, on average, the genes impacted by these rare germline structural variants had lower expression in general in the tumors, the matched tumors of these patients. Now there's a lot more noise that can go into measuring gene expression levels in tumors, a lot more factors to account for, but nonetheless, this is a significantly depleted expression level on average for the genes impacted by these rare germline structural variants. So to summarize what we found about genes in this study, we see that, like Riaz mentioned, genes that disrupt genes not only through loss of function, but also through putatively activating mechanisms like whole gene duplication or copy gain, are enriched in the germline genomes of children with Ewing sarcoma, neuroblastoma, osteosarcoma compared to controls. And these genes tend to not tolerate mutations in the general population, tend to encode certain subsets of proteins that we know already play roles in cancer pathogenesis, like DNA damage repair proteins. And there is some correlation that we probably want to dig into more in terms of where these genes are expressed, specifically in the tissue of origin for these diseases. But there's 98% of the genome that does not encode for proteins, of course. And so this is a tantalizing but pretty underexplored area, I think, in human genetics broadly, including in human germline cancer genetics. And so what we did here was we extended what Riaz did was he extended the category-wide association study approach that he enumerated a few slides back with an additional layer for non-coding and regulatory annotations. So these are things like histone modifications from ChIP-seq data on adult tissues. These are things like annotated cis-regulatory elements from various enhancer prediction elements. These are things like topologically associating domain boundaries, so on and so forth. And then focusing that analysis exclusively on structural variants that do not have a predicted gene disruptive or protein coding consequence. So these are non-coding rare structural variants being annotated and systematically compared between cases and controls for their potential consequences and or overlap with non-coding regulatory elements, putative regulatory elements in healthy adult tissue, not in tumors. And so these are the same volcano plots that you saw Riaz present from the coding CWAS, or the category-wide association study, except this time, this is only considering non-coding structural variants. And so a couple observations here are that, one, consistent with all the priors that we would have from other studies in non-coding germline variation, the effects of non-coding variants tended to be much weaker. So you can see that we don't have these, these towers of spikes in neuroblastoma. We do have a couple points that eclipse Bonferroni corrected significance for the total number of tests that we did here. But we see, no Bonferroni significant signal in Ewing's, which again matches what we saw in the coding variant analysis, where the signals in neuroblastoma tended to be stronger in terms of germline risk factors. But what did come of this analysis is, of the categories that eclipsed this multiply test corrected sort of Bonferroni, very strict Bonferroni significance threshold, was that they pointed us to singleton deletions in neuroblastoma that overlapped topologically associating domain boundaries defined, again, in adult adrenal gland tissue. So this is, again, mirroring the tissue of origin story that came out of the gene-specific work. And so there's a little schematic that we put together in the top right to sort of help contextualize the biological consequence of this. But essentially, what we are finding in a nutshell is that the rare deletions that are most strongly enriched in the non-coding region in neuroblastoma patients versus cancer-free controls tend to overlap the sort of choke point of these regulatory neighborhoods that inside the nucleus in individual cells in adrenal gland tissue cells. And this perhaps may have and has been shown in other contexts and other studies to have sort of a tack-on effect of disrupting gene regulation of the genes in these neighborhoods, in these domains. And one interesting finding that Riaz then pulled out in sort of a secondary analysis in the bottom right is that when we ask the exact same hypothesis in Ewing sarcoma for comparing to skeletal muscle regulatory domains, we see no evidence of a signal. And so this is really appears to be specific, at least from these results, to neuroblastoma and, again, adrenal gland tissue, not skeletal muscle for Ewing sarcoma. So just to sort of wrap up what we found in this study, I think there's really, at a high level, I think we've identified that rare germline structural variants, subsets of them are enriched in pediatric cancer cases over cancer-free controls. And so this may be an area where we can think about, additional risk factors and how best to define those for the context of screening and risk prediction. And then we also saw some trends that matched known epidemiology, such as the sex bias in the large rare structural variants, as well as the fact that neuroblastoma with a profoundly earlier age of onset than Ewing sarcoma tended to have stronger effects from these rare genetic variants, which is kind of consistent with what we would think about in terms of, not congenital disease, but very, very near, early life, first years of life. And the like. And so I think another sort of technical angle of this study that we thought was nice to see validated is that these types of population scale and genome-wide approaches can identify new risk factors that probably need further, more careful functional clinical follow-up, but nonetheless can provide new threads to pull on to help, expand the spectrum of risk factors for these diseases. And then just one other sort of note is Jeff McIntyre wrote a really kind sort of summary or perspective on this work that also came out in the same issue as our article. And they use this term inherited genome instability, which I think is really provocative and compelling. And we've also been talking about in parallel, how to interpret these large rare copy number variants that were enriched exclusively in males versus females, and some other signals like the DNA damage repair protein genes. And so I think this may be a really nice way to sort of like biologically summarize the overall top line finding, which is that these structural variants perhaps are conferring risk for these pediatric cancers through disrupting genome instability or genome stability in somatic cells, in somatic tissues. And so I think this is certainly very, very speculative at this point, but we're seeing some convergence between various different analyses that we do that all kind of lead us towards genome structure and the lack of stability in genome structure that might be sort of an underlying mechanism here that explains some of these signals.
So just to talk about some future directions, I think we're both very keen to continue to poke at this question of why pediatric cancer happens. And I think that there's a few different ways that we can continue to do this. So, we talked a lot about rare structural variants, but I think the other angle here was a global evaluation of genomes. And I think that there's still a lot of opportunity to do that with rare short variants. And so, again, with the genomes that we have, with genomes in the space more generally, I think that there's a lot more to unpack in terms of rare short variant risk factors when we expand beyond known cancer predisposition genes. Also, I think that there's a lot to explore here from the angle of constrained gene variants. So specifically variants, whether they be short variants or structural variants that are impacting mutationally constrained genes from a population genetic standpoint, and how these may be kind of new risk factors, especially in the context of the earliest onset cancers that we see. I think that, not something we went deep into today, but did kind of allude to in the context of the TAD boundary signals, just understanding germline epigenetics and the interplay with germline genetic risk factors, I think is a field with a lot of potential. And then, thinking about that spectrum that we started with of germline tumor, in a lot of the pediatric cancers that we see, translocations and rearrangements more generally characterize the somatic space. And so what are risk factors for these rearrangements and translocation in particular? And, I think the DNA damage signals certainly kind of give us clues, but I still do think that this is a question that needs to be answered. The focus of my lab is really to better understand what causes pediatric and young onset cancer so that we can better treat our patients. A lot of what we've talked about today squarely falls into aim one here of unpacking germline risk factors, but there's a lot that we're aiming to do on the tumor side as well, and understanding how the germline and tumor converge. And we're always eager to collaborate, and we're growing, and really looking forward to mentoring postdocs, grad students, and medical trainees. So certainly feel free to reach out if there's any interest in the communities that you all are a part of. So with that, we'd like to acknowledge mentors that made this work possible. So this project started when we were both postdocs in the Van Allen lab. And so a lot of gratitude just to Ellie, to the environment, and then to our other mentors listed here, to our colleagues and collaborators who made this work possible, and to our funding sources. And with that, we're happy to take any questions.
Well, thank you so much for that nice presentation. It's fascinating work, and I think opens up a lot of questions and new avenues for investigation. Anyone who's on the call, please feel free to write a question in the chat or unmute yourself, raise your hand, and ask a question. And I'll start off, I'm really interested in the predominance of the structural variation that you found in males. And I know some of that might be speculative, but do you have any ideas of what might be driving that, and where are you going to go next in terms of investigations to pull that thread further?
Sure. Yeah, so I think that's sort of one of the biggest glaring head scratchers that came out of this study. So I will say from a technical perspective, we did everything that we could think of to try and make that signal go away, and it really just does not, no matter which way we cross-analyze the data or filter. 98% of all the structural variants we validated through visualizing the raw sequencing data itself. Like these are real germline events. And they are, at least in the cohorts that we have analyzed, exclusively enriched in XY individuals. I think one important point that we did not mention was we also just asked whether these are large structural variants that just happen to disrupt known cancer genes. 94% of these large structural variants do not disrupt known cancer genes. So that leaves us with either they are acting through a gene that contributes perhaps some sex-specific bias, like perhaps this is differential usage of these genes in, somatic tissues in XY versus XX individuals, of which, transcriptomic population level studies have shown maybe 5% of genes can show some detectable, male-female bias in their expression levels. So perhaps these are landing on genes that are not recognized as known cancer genes, either in terms of predisposition or, somatic drivers. This is a pretty, comprehensive list of potential cancer genes that we excluded here. So they could be unknown genes acting through sex-specific pathway or mechanism, or sex-biased, I should say. It doesn't have to be specific. But I think one other observation one might make, and this is where we are going far into sort of speculative territory, is what genomically is different between XY and XX individuals is that the XY individuals have the added challenge in mitosis of segregating the large X chromosome from the small Y chromosome. And I should also mention that these large structural variants, virtually all of them appear on autosomes. These are not X chromosome structural variants. These are not Y chromosome structural variants. These are autosomal structural variants elsewhere on other chromosomes. And so perhaps it's the case that, XY cells already have the added mitotic stress of, segregating the X and the Y that are not the same size. Maybe these large, and again, this kind of lines up with the fact that these copy number variants are adding nucleotides or subtracting nucleotides from the overall length of one chromosome. And so perhaps it's the case that that is acting through sort of an extragenic mechanism of just like added mitotic stress or added mitotic challenge of now you have two sets of chromosomes in these XY cells that are very different very different sizes, at least in single-digit percentage differences. And so perhaps there's something going on there that might just make these individuals more predisposed to somatic genome instability due to mitotic non-disjunction, something like that. And then I think a third avenue that we did explore very provisionally is you could think of potentially imprinted loci. So differential methylation from mom versus dad, and what allele gets deleted. We, at a very cursory first glance, we saw no bias in terms of maternal versus paternal transmission of these variants in the affected children. And we also did not really see a lot of overlap with known imprinted loci. Perhaps there's unknown imprinted loci, but again, it's like the evidence is kind of getting pretty thin. And so I think we don't have a clear explanation, but we are very, very interested in figuring out what is going on there. And I think we're going to have to move to probably cell models, or more sort of molecular analyses to try and tease that apart.
Sure. And just a follow-up, I imagine you don't have this data, but there is also, are you able to hear me?
We're having a little bit of a hard time hearing you.
My computer does that sometimes. Can you hear me now?
Yes.
Yeah.
As a quick follow-up, I imagine you don't have this data available, but there is a slight increased risk of mortality among males compared to females. So I wonder if that effect is mediated at all by this increase in SVs.
Yeah, no, that's a great question. I mean, I think, the other, I guess, kind of computational angle for follow-up might be, regardless of mechanism, like, is this kind of a proxy for like pockets of variants more generally that seem to segregate in males for whatever reason? And I do wonder if there may be similar pockets in the short variant space. And so I think that'll be interesting to evaluate as well, whether rare kind of SNVs and indels seem to point to similar kind of male bias. Um, you cut out again.
How
We're
about
having a
now?
hard. Oh,
There you
yes,
go.
yep.
Okay. There is a question in the chat from Luis Sakamoto. Did you see any SV in microRNA or long non-coding RNA coding sequence regions?
Yeah, it's a great question. So our goal with the non-coding CWAS was to try to be exhaustive in a lot of ways and how we evaluated non-coding structural variants. And so just to go back to that framework over here, actually, I think I passed it. Uh, so it was a variation basically of this construct right here where we had additional layers for functional impact. And so even though we're trying to be less biased and more comprehensive, we did not actually evaluate overlap with things like long non-coding RNAs, pseudogenes, microRNAs. And so I think that like we're we're always kind of anchored in the biases of how we decide to look at the non-coding genome, whether it's kind of the functional regulatory elements or some of these other kind of very valid classes of non-coding genes that you mentioned. So I think it would be interesting to look at, but we just don't know.
Other questions from the audience?
Hi, can I ask a question? I'm in clinic. I hope you can hear me correctly. So I went on and off in the presentation and I apologize for that. I just want to know if the results you presented are mainly from European Caucasian populations and if there is any opportunity or to include other populations in the world that they may have either from an epidemiology standpoint, different distributions of cancer incidence. Like I was curious about neuroblastoma, which is the main solid tumor or second most common solid tumor in Caucasians. But in Latin America, where I'm from, retinoblastoma is actually way more common than neuroblastoma. And it's also a type of cancer from like little babies. So I just wonder if there is a chance to do that or if it was already done and if there's any results from that.
Yeah, sure. So I think this is we completely share the interest in expanding the patient catchment essentially of what demographics, both ancestral and histological, like what diseases and ancestries we can analyze here. I think this is a problem that sort of is currently plaguing in the field of human genetics more broadly. I know there's a lot of discussion about this in the context of GWAS for other diseases and so on and so forth. And I think practically speaking, we're as the New England Journal of Medicine would say, we are data parasites in that we are hamstrung by what data has been generated. We did not generate any of the sequencing data ourselves here. And so we can only work with sort of the patients that had the opportunity to be recruited into these studies, which is going to bias towards, relatively, Eurocentric and certainly also biases with socioeconomic status, access to medical care, so on and so forth. And so there's going to be some biases that we can't get around. I do think that we are particularly interested, and we share your interest in ancestry biases of, at the level of epidemiology, of different rates of cancers. So for instance, Ewing's is, if I understand correctly, is predominantly Caucasian. It appears in Caucasian children and teens. But then of course, like you point out, there's other histologies that, exhibit different biases. And so I think one direction that we are actively planning to build into as sort of a second phase of this overall effort of sort of genome-wide germline analysis is looking at some of those other histologies that are perhaps the ancestral distribution shifts to different ancestries or ethnicities. And so the real problem there will be identifying large populations of patients such that we would be sufficiently powered for those analyses from a statistical standpoint. Sample size is going to be a big limiting factor, but we are very, very interested in that as a next step of this overall effort to the extent that the data are possible to include.
And I think, one thing that's worth mentioning is, while there are kind of some ancestral tilts with some of these diseases like Ewing sarcoma, In this study, we, while not perfect, did have representation from non-European ancestries for about 30% of the population that we looked at. But I think that number needs to increase, and I think that, I also think that when we're looking in the context of specific ancestries and populations, like I think there may be opportunities to learn more about specific risk factors. So the example you bring up about retinoblastoma in Latin America is a very compelling one, and an opportunity, I think, to maybe go deep, with the right kind of planning to accrue kind of such a cohort over time.
Hi, I joined kind of late, so I only got the second half, but I had a question related to your ancestry comment. Since you mentioned you had 30% that was non-European, were you able to consider looking by ancestry to see if there were any differences? I know you might be underpowered, but definitely can't hurt.
And just to make sure we understand the question, you mean kind of specifically kind of like a stratified European only, or East Asian only, or kind of South Asian only enrichment analysis?
Yeah, I didn't know what the distribution of your ancestry looked like. So
yeah, so I think it was close to about 70% European, and then I think the other representative populations were about 12% African, and 12% admixed American, and then a smaller East Asian representation. So with that, I guess the ancestry stratified analyses kind of mostly focused on the most well-powered, which was European, like for sensitivity analyses for some of the signals that we were seeing, just to make sure that population stratification wasn't confounding some of the signals that we were observing. There may be, for focused questions, opportunities to go deeper and maybe some of the other represented populations, specifically the African and admixed American populations, where I think we had about 12% of cases coming from each. So it's a great question. We haven't done that in depth systematically for a lot of these signals. I suspect that just given the rarity of, the large structural variants, for instance, we would probably be underpowered for like that analysis. I think that it would be interesting maybe to take the top line findings that we saw from the coding or the non-coding CWAS studies to see if like some of those, maybe if we don't look in a genome-wide fashion, but if we try to replicate just in the ancestry setting on like a focused category, for instance, like are we able to do that? Yeah, it's a great question. I think an opportunity for going deeper.
The flip side of that too is one thing that we did in the study that's stuffed somewhere in the supplement is, not properly accounting for ancestry in these types of, multi-ancestry, large-scale association studies can risk pretty significant confounding from a statistical standpoint and lead to false positives. And so for most of the main like sort of headline results, the core results, potential associations and novel risk factors, we typically did do a sensitivity analysis restricting to only individuals of genetic European ancestry. So that rules out the potential for cryptic ancestral differences to be driving any of these signals. That totally does not get at the ancestry specific risk factor question that you raised, which I think is a really, really interesting one. But at bare minimum, we are well powered for a European only analysis, perhaps unsurprisingly. And so when that was relevant, we did that to rule out confounding from this multi-ancestry adjustment. But yeah, I think we're probably still a little underpowered, but it would be really interesting, like Riaz says, to look at maybe some of the top line results and just see if the effect sizes are different between different ancestries.
thank you.
Can I ask a question? Hi, I'm Miho Kato from Japan's Children's Cancer Group Data Center for Solid Tumors. Thank you very much for sharing great work with us. I'm building the long-term follow-up system for childhood cancer in Japan, And I'm very mindful of the implementation of precision medicine, as germline mutation can affect not only the risk of developing cancer, but also treatment related to long-term complications. So your purchase study suggested that the rare germline structural variants are risk factors for pediatric solid tumors. So would there be an optimal approach idea for childhood cancer survivors carrying those variants in terms of late complications, including secondary malignancies?
It's a great question. You know, I this question comes up a lot, and because it's such an important question, right? In terms of like this tie between the structural variants, the rare structural variants, and how they may be related to recurrence of cancer, secondary malignancy, or just outcomes more generally. And I think that like I guess two thoughts. One is I think the question generalizes beyond rare structural variants, and I think it kind of generalizes to just genetic risk factors more generally, right? And so I think that that still needs to be evaluated in the context of other rare variants that have been implicated in other studies. I do think that because, you know, on the one hand, we were able to benefit from what's in the public domain. It's great that this is out there, but what's still limited is the overlay of comprehensive clinical annotations. And so we don't often have outcome data for a lot of these patients. So it's hard to kind of evaluate that robustly. I do think that there are emerging cohorts where one may be able to look at this more comprehensively. So through the Children's Oncology group, there's a study called MATCH that is actually focused on kind of relapse refractory cancers. And so I think there's a pretty sizable cohort, not histology specific, but across a lot of extracranial solid tumors and then also brain tumors, where there is a good amount of outcome data that is going to be available, where I think, you know, one may start to ask questions about germline genetic risk factors, structural variant and otherwise, that are associated with different outcomes. I also think that, you know, even with these cohorts, you know, some of them, a lot of these samples came from St. Jude. So that data's somewhere. We just have to try to get to it. And then a lot of these samples came from Gabriella Miller Kitts First, which ultimately came from the Children's Oncology group. So like that clinical data is somewhere. We just have to get to it. But you know, it's surprisingly sometimes it's a challenge to get to that data. And I think it's a really important question that you're asking.
If I could just piggyback on that too, from taking sort of the more population genetics standpoint, I'm not an oncologist by training. I'm a scientist purely, and so I would say that a lot of the signals that we see here are not specific to pediatric cancers. In that, so some of the top line findings are disruption of genes that are highly expressed, disruption of mutationally constrained genes. These are genes that we know are technically speaking pleiotropic, in that they confer risk for a variety of different diseases, of which now pediatric cancer is a you know, known component of that spectrum. But so I think that does provide an opportunity. And like I mentioned earlier, the large rare structural variants are very, very important in pediatric neurodevelopment. And so I think knowing that these risk factors are now associated with pediatric cancer allows us to ask questions about, okay, well, what is the risk for X or Y disease that may have already been studied sort of in the population level of, you know, biobank studies, UK Biobank, All of Us Biobank, Biobank Japan. Other groups have looked at the association between such rare variants, rare structural variants, rare coding variants in impacting these same genes. And so perhaps that's an opportunity to say, to essentially bootstrap, if you will, from, oh, this structural variant impacting gene A is, you know, associated with pediatric cancer X or neuroblastoma, but it is also known to have an increased risk of, you know, early onset heart attack, or so, just totally spitballing. But that illustrates the opportunity to use these biobank scale data sets to sort of connect the dots between maybe, like you say, sort of secondary complications later in life that might derive some risk from the same genetic underpinning.
Thank you very much.
Thanks to both of you again for the presentation and stimulating discussion. I just have one more suggestion comment. As I'm sure you know, that birth defects are highly associated with childhood cancers. It may be interesting to see if any of the genes that are impacted by the variants you discovered are known to be associated with structural birth defects as a future direction.
Especially with the constrained genes and the variants that we're seeing there, I think that there is probably a lot of pleiotropy, like you said, and I think a lot to kind of uncover there. So yeah, that is a fantastic suggestion. And then I think this came through as a direct message, so I'm not sure if everyone can see it, but one question from Stephen, he's a medical resident in genetics in Columbia. Do you recommend performing somatic and germline studies in patients with osteosarcoma for hereditary cancer risks? So I guess, I think that genomes, both tumor and normal, are helpful in terms of characterizing biology. I think that in terms of understanding hereditary cancer risk, I think the more genomes we have, the better. And so I think that's specific to osteosarcoma and more general as well. I think we still have to kind of unpack this, like where we're looking and how we're looking, but I think, better power will help with that. So I guess the short answer is yes to that question.
Okay, well, thank you so much again, and thank you everyone for the discussion and the questions. It was a pleasure to hear about your work, and I hope everyone has a wonderful rest of their day or evening, wherever you are in the world. Thank you.
Well, thanks so much.
Thank you.
Bye.